This lecture is going to talk about an aspect of micro in preventative medicine that's critically important, and that is vaccinology and vaccine development. So this lecture is going to consist of three major topics. The first is to just clarify what we mean by immunization versus vaccination, because they're not strictly speaking interchangeable. We're going to also spend most of this talking about different types of vaccines. Not only how you deliver them and what the appropriate schedules and groupings are, but the biological differences between each type and what their risks, benefits, etc. are. I've color-coded these to make things easier. So each time we're talking about a subunit vaccine, you'll see it labeled in red. Each time we're talking about a toxoid vaccine, you'll see it labeled in green. You do need to know what vaccine is what type. That's critically important. And the way that you're going to piece that together from this class material is by the color coding. Finally, I'm going to talk very briefly about resurgence of vaccine preventable diseases and a few notorious examples thereof. All right, so first things first. Immunization in its most literal terms is the creation of immunity, and that can be done actively or passively. And if it's done actively, that means your own body or a patient's own body is generating the immune response. If it's done passively, it means that somebody else has generated that immune response and that the patient in question is simply receiving the benefit of that immunity. That can be done naturally or that can be done medicinally. But the key difference between the two aspects of immunity is that whether or not the patient themselves is actually generating the immune response. In the case of active immunization, the patient will have a set of memory cells develop. In the case of passive immunity, they will not. So in any event, active immunization is always better. However, it's not always clinically possible to generate in a timely fashion for the patient. So you'll see a great example of that in a second for what we mean by that. So that being said, immunization is not strictly speaking interchangeable with the term vaccination. Vaccinations are always immunizations. Immunization is not always a vaccination. Make sense? Okay. The example that I mentioned a second ago that we wanted to bring up at this point is rabies. So there are two clinical rabies immunizations, and one is active and one is passive. The active immunization for rabies is a killed vaccine. You give it to somebody prophylactically so that they can generate an immune response to rabies. The passive form of rabies immunization is purified human IgG, reactive against rabies virus. In this case, you give this to somebody who's got a high risk of exposure to rabies, somebody like a veterinarian. In this case, you give this to somebody who has been bitten by an animal and you suspect that they have had rabies exposure. The reason why you treat this differently and why you care about creating lifelong immunity in this patient, but it's not your first line of action for this patient, is that you want those rabies viruses, if any were injected, to be immediately sopped up by antibodies in a person who has no pre-existing immunity. By the time a person effectively generates an immune response that's going to clean up enough rabies virus, you risk the possibility that that virus has already entered the central nervous system, wherein a circulating antibody response will not be nearly as effective at elimination. And if you did have adsorption of antibodies in the brain, in the CNS, then you risk having a lot of damage to the CNS. So it's critically important here that you don't try to stimulate active immunity, that you immediately give passive immunity. after which, mind you, you also give them this afterwards. It's also reasonable to point out that you want to give the active vaccination to somebody who has, again, repeat risk of exposure through their occupation or where they live geographically, whatever it is, rather than giving them passively transferred immunity. If you know that contact with that agent is probable, then it's probably best to make sure that person has their own active response and their own memory cells on board. All right, so I like this particular graph because this particular graphic because it's a nifty little sort of faux phylogenetic tree of the types of immunization. So by and large, you can divide it into two major categories: passive here and active here. When we look at passive, we look at the transfer of serum immunoglobulins, specific IgGs or monoclonal IgGs. Here we've broken this into the two types that are medicinally used in humans, which is recombinant human IgG or equine IgG. This is rabies, this is tetanus. Monoclonal antibodies, which I believe you would have discussed with Dr. Sau or Dr. Morganelli, these are where you have a completely clonal population of antibodies that are only going to recognize one specific epitope. There are a few approved therapeutics that are monoclonal antibody based, but mostly they target cancers. Most notably, the breast cancer medication Herceptin is a monoclonal antibody. Specific immunoglobulins is sort of a vague term, but how I would interpret that would be to look at some of the natural ways that immunity is passively transferred. So in a second, I'm going to say pause, and I want you to think about what that could possibly refer to. Okay, pause and think. All right, unpause. What that refers to almost exclusively is breastfeeding or transplacental transmission of IgG. Okay, so now to move down to active immunity, we are looking at live immunizations, inactivated immunizations, or subunit immunizations. So inactivated forms are where you're looking at a killed pathogen in its entirety, certain pieces of it, whether they be peptides, polysaccharides, or toxoids. Those are the big ones that we want to be familiar with. And then down here, you can generate active immunity by interacting with live pathogens. Most of these, when you're talking about immunizations, are attenuated mutants. So these can be attenuated in multiple ways. We'll talk about those in a second, but they're basically live, almost exclusively virus, and these viruses are attenuated to the point where they can't cause disease anymore, but they're quite good at stimulating protective immunity against fully virulent forms of that virus. Hybrid viruses are what I refer to later on as recombinant vaccines. There's one really promising example of that we'll talk about in a few minutes. And then finally, the other way of obtaining active immunity that we want to kind of recognize as a thing is this. If you have active immunization with a virulent strain, that's a really fancy way of saying you got sick, you developed an immune response, and then you got better. The next few slides, we're going to talk about each of these different types of vaccines in greater detail. After that is when we're going to talk about different target populations for each vaccine, and then you'll get to see which infectious agents are protected by which type of vaccine. This is also where this color coding starts. So anytime you see black and white, you are looking at a whole organism vaccine. Whole organism vaccines are going to fall into two different categories. The first is live and the second is inactivated. So these are living things, these are dead things. As mentioned, live vaccines are always attenuated. They can be attenuated in multiple ways. The first thing I would point out is that there's a huge aspect of cell culture here. Some of you may have done this if you've worked in a laboratory setting, but if not, basically what cell culture entails is growing vertebrate cells. Actually, that's not strictly speaking true because there are insect cell lines. This involves growing eukaryotic cells that are not yeast in a flask. And what you do when you do that is that you have these guys transformed in a way. They're not normal standard cells because normal standard cells do not like to grow close and snug to each other in a dish. They have something called contact inhibition, and they like to grow in three-dimensional spaces into tissues. So what you could take away from that is that cell culture lines that have been adapted to grow in a laboratory are not quite biologically normal. Okay, so that said, when you take certain infectious agents, such as a lot of the childhood viruses, measles, mumps, rubella, varicella zoster, and you adapt them to grow in these abnormal cells, what you're doing is a twofold benefit. First of all, you're developing a system to be able to grow a whole bunch of them in a laboratory so that you can then purify them and make them into a vaccine. But more importantly, what you're doing is you're adapting them to these abnormal cells, and in so doing, you're adapting them away from normal cells. Okay? Does that make sense? So when you condition these guys to grow very efficiently in a laboratory, you're simultaneously making it so that they don't do quite as well in a live human being. In other words, you're attenuating them. In addition to that, and I would put this kind of as a subheading, there's also a trick called cold adaptation. And how this works is that you propagate cell lines that you're going to use to grow viruses at temperatures that are lower than human body temperature. Typically, that would be 34 degrees C. When you get viruses that you've selected for and adapted to grow at that lower temperature in these abnormal cells, when you put that back into a human body, which has normal cells and is at 37 degrees C, that virus is not very happy there. It doesn't do super great. So in this regard, it's yet another addendum in the ways that you can adapt these guys and attenuate them. The final thing I would point out here is that you can also chemically mutagenize pathogens. And in this way, you can attenuate them. You introduce so many base changes all at once that you end up knocking out some of their key virulence factors. So that being said, this trick here is not used all that often in human medicine. It's used more in veterinary medicine, but it is a very effective method of attenuating certain strains. Okay, so live whole organism vaccines have a lot of benefits and they have a few risks. First of all, the benefit received from these vaccines is that they are far and away the best simulation of naturally acquired immunity. It's very difficult to find strains that can escape vaccine protection. It's not impossible, but it's difficult on the whole. They also have the benefit of creating a more relevant immune response. So you, in this regard, are going to have T and B cell responses. You're going to have if it's relevant, you're going to have, say, secretory IgA responses in addition to just IgG responses. It's, again, the best way of putting it is it's the single best simulation for naturally acquired immunity. However, that also comes with a few risks, and the first is that if you have immunocompromised household members, and that can be an elderly person, that can be a person living, a sibling with an acquired immune deficiency, could be an HIV patient who lives in the household. But when particularly a child is immunized with a live vaccine, they do shed infectious agents. They are attenuated. Most of the time that is not going to cause anyone any kind of problem. However, it is something that you have to remember if you have an immunocompromised person living in the household. In addition to that, there is also the remote potential that you can have a reversion to virulence. The best example of this happened with oral polio vaccine strains. Now, in the United States, we no longer use the oral polio vaccine, but that was a decision that was made not so very long ago. I myself had the oral polio vaccine as a kid. At this point, we use an inactivated one. We'll get back to those in a second. But the oral strain, while it provided superior protection, it did have this very, very, very low risk of reversion. And so it got to the point where polio in most of the world was sufficiently under enough public health control that that very small risk of reversion became unacceptable in comparison to the risk of infection. And so most places in the world have made the decision to switch back to the inactivated vaccine. So speaking of which, inactivated vaccines are whole organism, same as live attenuated. However, they are simply chemically inactivated. When you ask how that is, it's usually by treating them with formaldehyde. After which the formaldehyde is clearly washed away, completely removed from the system until you have only very trace amounts that are well below acceptable exposure limits. And these agents are completely dead. They're dead as a doornail. I'm emphasizing, let me write it down to make sure we're all clear on this. Dead, with a big skull and crossbones under it. I'm emphasizing this because there are a lot of misconceptions about certain vaccinations, particularly with how it applies to shedding and creation of the disease that they're intended to prevent. So when people talk about shedding of pertussis vaccines, not possible. These are inactivated previously, and now they're subunit vaccines. Back to what those are in a second. But it's critically important that we know which vaccines are inactivated, and that we're all very familiar with that, because when we need to counsel patients about what their concerns are, when many people bring up shedding and risk of active disease, it's very helpful, I think, to be able to say to a patient that isn't possible because those agents are dead. They're killed. So the risks of these are very, very minimal because, in case we haven't flogged it enough, they're dead. They cannot cause the disease that they're intended to prevent. So the risks of these are essentially only what you'd find in the risk of giving an immunization at all, which is, you might have some aches and some low-grade fevers and, maybe a bit of crabbiness or an upset stomach for a day or two, but the risk of anything more serious than that is extraordinarily non-existent. The benefits, I apologize for that. The benefits are that you have no risk of infection. So these are quite safe. The problems with them is that they don't work as well. I don't know how to write that other than a sad face in that space, but they don't work nearly as well as live vaccines. They're infinitely better than acquiring immunity the old-fashioned way by getting sick, but they're not as immunogenic, and it's easier for pathogens to escape the vaccine. A great example of this is that we all require an annual flu shot. Most of the flu shots that we get as adults are inactivated. Our flu shot can't adapt. It can't. It can't be shifty or drifty in any way. It can't change. And so we're going to make immunity solely and only to the thing we were injected with. Not that live flu shots do a whole lot of adapting, but they tend to be a little more heterogeneous than inactivated ones. So that being said, our immune response is quite limited to what we were injected with. The next group of vaccines we want to talk about are subunit vaccines. So the major difference between whole infectious agent vaccines and subunit vaccines is that subunits contain only a single part of the infectious agent, not the entire thing killed, not the entire thing live, certainly, but only a certain part. So that being said, subunit vaccines tend to be smartly designed. So a lot of people have done a lot of work with molecular biology in the past few decades, and a big part of that has been looking at sequences and looking at very distinct biochemistry of a lot of different things, and infectious agents are no exception. One of the key things that people have done when they were looking at sequencing of pathogens was to look at the protein sequence, and this would be exemplified by this string of letters up here, and trying to find which parts of this protein sequence would be most immunogenic. So in other words, would be most likely to stimulate an immune response that is protective against that pathogen. So you're looking at two different things. You look at the kind of the biochemistry of that little epitope right there to say, all right, yeah, that looks like it would be quite flashy and the immune response would pick right up on it. Just as a hint, that has a lot to do with the hydrophilicity of the epitope. And in addition to that, you have to make sure that that particular string of amino acids is going to generate an immune response that's protective. And so that being said, these peptide vaccines have been attempted to be developed for a few different things. One is still on the market. It's highly safe. It's highly successful. One was pulled from the market, but it was quite effective. It had a high side effect profile, but it was quite effective. The hepatitis B vaccine is the most commonly used peptide vaccine to this day. It's worked extraordinarily well, and again, it's extremely, extremely safe because it does not have whole virus. The production of this vaccine involves synthesizing a peptide off of a machine, cleaning that up, resuspending it, and then using that for injection. So there's no virus anywhere in that process. There's no live virus grown up and injected. There's no virus grown up and then killed where something could maybe go wrong in that process. It's completely and utterly synthetic. And so there is absolutely no possibility of ever contracting hepatitis B from this vaccine. So its benefits are that it's very, very safe. It's very, very effective. So that's fabulous. The risks with peptide vaccines in general is that you can have escape. Hep B has thus far not escaped its vaccine, which is great. But it is quite possible that if you have an infectious agent that evolves quickly and you're only making epitopes to this very antibody right here, I believe I said that backwards. If you're only making antibodies to this epitope right here, if you get one or two changes in there, you're going to have a strain that escapes your vaccine. So escape is a big risk, but with our one that we use the most right now, it hasn't happened yet in about 20 years or so. So that's pretty promising. The other peptide vaccine that was in use was something called LymeVax, and this was protective against Lyme disease. About 5% of the population seemed to have a long-term side effect of arthritis, and the reason for that was that the chosen epitope for LymeVax seemed to cross-react with self-antigen from a very small proportion of the population. And at the time, Lyme disease was not so prevalent that as it is now, and so that was seen as an unacceptable level of risk of side effects versus the risk of infection. However, we all might, in this day and age, want to rethink that a bit, or at the very least, think about redesigning that peptide, because the actual vaccine itself for protection worked quite well. Okay, so those are peptide vaccines. The other infamous subunit vaccines are polysaccharide vaccines. I trust you talked about capsules in week 4 or week 6 with Dr. Lee, but in case we need a refresher, polysaccharide capsules are this kind of layer of goo that sit around the outside of certain species of bacterial cells. So this right here would be a single cell. It's in the process of dividing. All this gray jazz surrounding it are the capsular polysaccharides. So it turns out for many bacteria, these particular linkages of carbohydrates and the proteins they're stuck onto are quite immunogenic. And so if we have a good understanding of which pathogen has what subtypes and what polysaccharides, we can do very much the way we did with peptide vaccines, design something that will be both immunogenic and protective against certain bacterial pathogens, and we can synthesize those in a laboratory. The same exact principles apply, as we said, with peptide vaccines. There are no infectious agents as part of the process of making these vaccines. So it is not possible to contract the disease from the vaccine. It's got an incredibly good safety profile. So we'll put that under benefits. They tend to be somewhat effective with a caveat. They do work extremely well. However, pathogens that have polysaccharide capsules and have very immunogenic capsular antigens tend to be very good at varying those antigens. So, when you generate a vaccine against capsular polysaccharides, they are effective. However, some of these agents are extremely good at escaping these vaccines. I believe you learned in week 4 that there is, and has been for several years, an available pneumococcal pneumonia vaccine, which there is. It is a polysaccharide vaccine. It's got a mixture of 23 different capsular antigens in it that cover 23 different strains of pneumococcus. The problem with that is that there are a lot more than 23. So all of the other strains that are not protected by that vaccine are still quite able to infect and cause disease in patients, even though they've been vaccinated. So it's very important to appreciate the nuance here. It's not that these vaccines don't work. They work quite well. They just work very, very specifically, only against certain strains. So it's important when we're talking with patients to make sure that we recognize and that we're able to communicate to them, this works very nicely, however, it doesn't work completely. So the best way and the most straightforward way of communicating the situation with these vaccines would be to say that receiving the vaccine is going to greatly reduce their risk of developing pneumococcal infections, for example. However, it doesn't eliminate it completely. And the reason for that, once again, is that it's going to eliminate the possibility of any of those 23 strains infecting a patient. It is going to do very little for any other strain. Make sense? All right, on a related note, there's another group of vaccines called toxoid vaccines. These are technically, to me, I would consider them to fall under the category of subunit vaccine, even though they were named first and they were invented first. These guys are not designed the way that peptide or polysaccharide vaccines are. These guys are developed by looking at diseases that are caused exclusively and only by toxins, by exotoxins specifically. So the biggies for that would be tetanus, diphtheria, and historically pertussis. It's not that pertussis was historically caused by toxin and now it's not. It's that we historically had exclusively a toxoid vaccine and now we combine it with something else. More on that in a minute. So these guys are made in something of a more crude way. In this case, you take the toxin and only the toxin, purify it, and then formalin fix it. When you do this, in other words, formalin fixation is a fancy way of saying I threw a bunch of formaldehyde at it. When this is done, you'll notice there is no inclusion of the infectious agent here. So there aren't Corynebacterium diphtheriae cells in that suspension. There are not Clostridium tetani cells in that suspension, nor are there Bordetella pertussis cells in this particular suspension. We've got this combination vaccine I'll get back to in a moment, but in this case, we are only taking the toxin because the toxin is the thing that actually creates the disease. The bacteria themselves are kind of incidental to the whole thing. We take toxin, when we formalin fix it, you take away its ability to actively mediate disease. However, most of the epitopes on that toxin are still intact. And so a patient is going to be able to mount an immune response that would come in and neutralize that toxin before it's able to actually go in and create cellular damage and cause disease. So this is where we start getting into the realm of upcoming vaccines. The next two that I'm going to talk about are very conceptually newer than some of the more classic vaccines. They're not, I would say, new at this point, but we don't have a lot of commercially available vaccines that fall into any of these categories. There are some upcoming, and I'll talk about those in a little while. So basically, I lost my arrows here. The first of these that I'm going to talk about are called DNA vaccines. This is not a vaccine that is raising an immune response to pathogen DNA. These are vaccines that are made of DNA. Okay? That's a point that trips some people up. So I want to make sure we're very clear on that. Okay. When you're generating a DNA vaccine, you want to start in the same exact place we did for peptide vaccines or polysaccharide vaccines, and that's by selecting an antigen. Looking at a pathogen's genome and saying, that right there sits on the surface, it looks very immunogenic, and it looks like when I screen patient antiserum, it looks like there's protected, there are antibodies present to that antigen that mediate protection. So you select your antigen based on those factors, and then you clone it into a piece of plasmid DNA. So in this diagram up here, this would be a gene that encodes your antigen, and this would be the act of cloning that gene into a plasmid. So what you have right here is a self-replicating little piece of DNA, and this DNA is going to be able to express this gene right here that you cloned in and make antigen in whatever cell you put it in. Okay. At this point, once we have this plasmid generated, it's injected into patients, and it's usually, there's a picture of a needle here, but it's usually done by a device called a gene gun. Basically, bits of DNA are coupled to colloidal gold, and then that colloidal gold is more or less air blasted into patient's tissue. Usually on the upper arm, sometimes on the thigh, is where the studies have been done. And what happens here is that you have that bit of DNA injected directly into patient cells. The patient cells start reading this and expressing this gene. And then those patient cells start creating that antigenic protein. The antigenic protein, even though it's made by patient cells, is non-self because it's a protein that comes from a pathogen. And so your immune system, or a patient's immune system, is going to start generating immunity to that, just as it would any other non-self antigen. As you could imagine, this is completely devoid of pathogens. There's no infectious agent in that syringe or that gene gun. One of the issues that has come up historically with DNA vaccines is that they tended to work very well in theory in the laboratory, but when you actually put them into animal trials and in some cases into human trials, they tended to not work super well. And the reason seemed to be that expressing these genes didn't happen at a very high level until very recently. There have been a few developments now that have really seemed to have overcome that problem. And so there are a couple of vaccines that are currently in clinical trials that are DNA based, that seem to be working very, very well. Okay, so the final vaccine type that I want to talk about are recombinant vaccines. So these are very much like DNA vaccines, quite novel in nature. They're very smartly designed, so to speak, as opposed to the old way of making vaccines, which is sometimes colloquially referred to as kill it and fill it. So these are going to work very much the same way, where you select an antigen based on those same factors. You clone it into a delivery organism, or you replace the delivery organism's analogous gene. And what we mean by a delivery organism is that you can take either an attenuated or a harmless virus or a probiotic bacterial cell, and you can get them to express this pathogenic antigen, or this pathogen's antigen. The two that are used most commonly are a virus called vesicular stomatitis virus, and this is a murine pathogen. This does causes no disease in humans whatsoever. I've also read quite a bit about people trying to use the human commensal lactobacillus. However, the only recombinant vaccine that has been approved for use in humans, and it's approved for use in Mexico, it's not yet approved in the US, is using a backbone of vesicular stomatitis virus. Another that was put into rapid review clinical trials seems to work beautifully well, also uses VSV as a background. We'll show you what that is on the kind of upcoming vaccines slide in a few minutes. But anyway, the gist of this is that you have your delivery organism, you have your chosen antigen, represented by this kind of red gene against a blue backdrop, and you make what's called a recombinant viral vector, or bacterial if that's what you're using. But in this case, if we're talking about vesicular stomatitis virus, we're going to be looking at a viral vector. And basically what you wind up with is a virus that's chimeric in a way. And it'll have a genome that is mostly VSV and just has this one gene from some pathogen of interest. And so what you wind up with, again, are these chimeras over here, where they're essentially VSV, but then they have this one antigen on their surface. And what happens when you use a recombinant vaccine like this is that you get the benefit of having a live vaccine. So all those things we talked about in terms of kind of this mimicry of a natural immune response, you have that and you get that with these recombinant viruses. However, you do also have the limitation that you see with some subunit vaccines of things like escape, because this is an artificial thing. It doesn't occur this way in nature, and so this is not going to change. This is going to be this one version that you generated in the laboratory, and that's the only set of epitopes that a patient is going to mount an immune response to. Any strains that escape that immunity are going to still be able to infect. Okay, so I want to talk briefly about vaccine complications. By and large, the overwhelming majority of the side effects from vaccinations are low-grade fever and injection site pain. Something that I hadn't mentioned as yet is the concept called adjuvancy. Sorry, that's a U, that's a V. Adjuvants are compounds that are added to vaccinations to be more stimulatory to the immune response. The benefit of using either dead things or synthetic things is that there's no risk of infection. However, the problem with that is that they are not nearly as immunogenic as living things are. And so in order to get around that, you use an adjuvant. So these are things like alum or things like oil, and these guys are just going to stimulate nonspecific immune responses, and they're very, very pro-inflammatory. The problem with that, or the complication with that, is that they tend to give you fever and pain. Anyone who's had a tetanus shot can attest to that. They're extraordinarily painful. You tend to have an achy arm for a few days after that. And that has everything to do not with the toxoid, but with the alum in the shot. That being said, one of the things that is rather reassuring about this fever and pain response is that it's working. This is what you've done, is create a very potent and strong immune response, and that's what you want to do. If you don't create an immune response, the vaccine has not done its job, right? So another minor complication that you sometimes see is a rash at the injection site, and you particularly see this with live attenuated vaccines, such as measles, such as chickenpox. These go away in a few days. These also tend to go away in a few days. They can be readily controlled, obviously, with NSAIDs. Very rarely do you see anything more complicated than that. When you do, it tends to be if you have a patient who's allergic to a certain component. A notorious one is the influenza vaccine is grown in embryonated chicken eggs. And so patients who have egg allergies can sometimes react poorly to the influenza vaccine. Okay, but by and large, the two complications that I want to talk about are disease contraction and failure to protect. It is possible to contract a disease from an immunization. So we already briefly mentioned the oral polio vaccine, that was on very rare occasions giving people mild cases of polio, until it became such that it was not an acceptable risk, and we started using the IPV instead. So that's one issue. There have been reports of mild cases of chickenpox following varicella-zoster vaccine immunization, but that's about it. So that being said, and these are in immunocompetent patients. In immunocompromised patients, this becomes a little more complicated, and it becomes a little more plausible that an immunocompromised person can contract a disease. However, the one thing I do want to bring up is this sentence right here. I got the flu from my flu shot, and that sentence drives me bonkers. And the reason it drives me bonkers is that, again, most adults get an inactivated, killed flu shot vaccine. So by and large, what ends up happening is that there's a misconception here. And it's really important that we be able to communicate with our patients about this, because flu shots are extraordinarily important to get year after year. 30,000 people per year have severe or terminal complications from influenza. That's not an acceptable number to me when you consider how few people get their annual flu shots. It's something that we don't think of as a big deal because you hear about flu all the time, but it's an incredibly, extraordinarily dangerous infection. And it's vaccine preventable infection. So there are, I think, 3 elements that go into I got the flu from my flu shot. And the first goes right back to these complications of fever and pain. Sometimes people mistake the side effects for flu-like symptoms. Like, I got the flu shot, and then, like, later that night, I started not feeling good, and I got a fever, and I got the flu, and it was the worst. And then they say, yeah, 8 hours later, I was fine. You're like, yeah, because that was the side effects of the vaccine. You didn't have the flu. So one issue here is side effects. The second is that as we learned a couple weeks ago in week 6, there are many things that cause influenza-like illnesses. Influenza obviously is among those, but it is possible to have a flu shot 2 days later come down with RSV, or come down with the coronavirus infection, or come down with West Nile infection if you get your flu shot in late summer, and then conclude that shot gave you the flu, and it just was a terrible coincidence in timing. The third is that it takes about 2 weeks to generate full protection, and that kind of mimics the curve of adaptive immunity, right? You got about 10 to 12 days before you start getting nice antibody responses. And if you happen to get the flu within those 2 weeks until you're fully protected, then that would more or less give you the idea that you either got the flu from your shot or your shot didn't work. So now we want to come right down and segue into this failure to protect issue. And a lot of people go into this sentence: I got a shot and I got the flu anyway. So that point that we just made up here plays right into that idea of if you got the flu 3 or 4 days after your flu shot, it's not that your shot wouldn't have worked, it's that you happened to get sick before it was fully effective. That's one issue. The other has to do with the word flu, and that phrase that you all know that I hate, stomach flu. That's not a thing, as we discussed during the virology week. I've had people say to me, I got the stomach flu, which is really annoying because I got my flu shot and you said it would work. Which, of course, the answer to that is, quote, stomach flu is not caused by influenza virus, which is what flu shots protect you against. It's caused typically by norovirus or Aichi virus or something like that. And so there's a misconception that these shots don't work. The problem, of course, is a little more complicated than that. But I just want to pause for this moment before I talk about this point down here and just say all of these reasons we've just been discussing are why a lot of people do not get flu shots. And it's critically important that we are able to communicate with patients who are asking questions why we have these misconceptions and where they come from. And it's just, it's not acceptable that we have as poor an influenza vaccination rate as we do, considering how deadly and consequential this infection can be, not just for your patients themselves, but if they happen to have, elderly relatives or relatives who are infants or immunocompromised relatives. It's really quite, or sorry, I should say asthmatic relatives as well. It's really not a trivial illness, and the vaccine has a lot of, it's not perfect by any stretch, but it's not got the problems that people suggest that it does. So it's critical that we're able to talk persuasively about this and articulate where those misconceptions come from. We don't tell our patients they're wrong, mind you. We just have to be able to communicate why these perceived problems exist and what we can do about them and why it's still a good idea to get the vaccine anyway. Okay, so under the heading of failure to protect, there's this issue of prevention of disease versus duration and severity of disease. As much as I just spent time saying yay flu shots, they are not completely protective because influenza is a very diverse virus. And we make our best guess at what strains are going to be circulating in any given flu season, but we're not always right. And sometimes we're extremely wrong, or sometimes a brand new strain emerges and catches everybody by surprise. And so, once again, we generate immunity to the thing that we are injected with. And if we have strains circulating that escape that immunity, then that vaccine is not going to be particularly protective against them. That's also why we change the flu shot from year to year to year. As these viruses change and adapt, we also change and adapt the makeup of the immunization. But that being said, It certainly isn't the only vaccine that warrants discussion here. Duration and severity is a really important point. You can have incomplete protection against certain infectious agents, and it's infinitely better than no protection at all. Because if it may not prevent you from getting the disease, if it makes it so that you have a milder case for a shorter amount of time, that's still good. One of the best examples of this is the shingles vaccine. It's only about 65 to 70% effective in preventing shingles in elderly patients. However, the patients who develop disease, it's invariably shorter and less severe. It's certainly not the only example of that. But that being said, if you have patients that are saying, well, I heard this doesn't always work, that's a really important thing to discuss with them. It may not always prevent completely, but if you can cut it back and make it less painful and less serious, that's absolutely worth doing. All right, so quick point about vaccine administration. Vaccines can be administered in one of 4 major ways for humans. For veterinary medicine, we have a few additional ones, but far and away the most common is intramuscular, meaning you do a quick injection into the muscle tissue. Intranasal has been developed reasonably recently. The big one that we see here is FluMist. So this is the live version of the flu vaccine. The intramuscular version is the killed one. This is often given to smaller children that are perhaps a bit needle averse. Oral vaccines, there's quite a few of those. We mentioned oral polio. This would be the 3rd time now, but we don't really use that anymore. There are 2 oral vaccines used quite common, or one used very commonly and one used for only certain circumstances in the U.S., and those are the rotavirus vaccine and at least one of the typhoid vaccines. And then gene gun is how you typically administer DNA vaccines. Again, we don't have any of these approved at the moment, but we have 2 in clinical trials that seem to be working reasonably well. One other point to bring up is that as much as we talk about individual disease vaccines, in many instances what we have available commercially are what's called polyvalent vaccines, and that means that, and so I've got a package of one right down here where we've mixed meningococcus and the diphtheria toxoid vaccine. We look at combination vaccines where we put multiple vaccinations together in the same injection vial and into the same formulation. And this reduces costs. It cuts down the number of immunizations that any individual has to take. And in addition to that, what ended up being a kind of an unexpected bonus to this is that when you put multiple infectious agents together, sometimes they act as each other's adjuvant. And so the neat part about that is that you can almost cut down some of the chemical adjuvants and reduce the side effect profile. One of the issues with elective omissions of vaccines or these kind of advocated alternative schedules is that sometimes people are not fond of this idea of polyvalent vaccines. And the argument that's given is that, children are given too much too soon. There's too many antigens in this. And the problem with that is that, first of all, I understand where that would come from, in that you take a small child and you see 5 or 6 needles lined up to shoot things into them, and you learn that one of those needles has 5 different vaccines for 5 different infectious agents in it, and it's alarming. It's alarming to parents, and the issue though is that it's a matter of perspective. We can physically see those vaccines, and we see them being injected in, and we know how many infectious agents are in them. However, I think it's important that we give context to this. And the reason and the way that I think it's best to give context to this is to tell people about your reaction when I say the following. Imagine for a moment you take your hand and you stick it on the railing at Disney World. And then you lift up your hand and you eat a burger with it, or you lick it, because let's pretend for a moment you're a 2-year-old toddler and that's what you do. You lick things. The number of antigens you have just introduced into your body is far and away more, like orders of magnitude more than you are getting in a vaccine. Hopefully that gave you a little gut punch, and be sure to share that with your patients. Again, it's important that we sympathize with how these immunizations look to parents in particular, but this perspective is critically important to communicate. All right, so who gets which vaccines? And here's where our color coding kind of comes into play. The first thing I'm going to discuss with you is the childhood series. And starting at a 2-month visit, these are the recommended vaccines for a child in the U.S. Our live attenuateds that we get are Rota, BZV. Our subunits are pneumococcus and HIB, that stands for Haemophilus influenza B. This is a bacterial species that causes ear infections and meningitis. This guy causes, in small infants, typically causes ear infections. In older adults, in adults, it'll cause ear and sinus infections, and in the elderly, it'll typically cause pneumonia. Sorry, I flew right by it, but rotavirus causes just a viral gastroenteritis, a vomiting, diarrhea, dehydration, nuisancy kind of issue. For our killed vaccines, we have the inactivated polio vaccine. I have pertussis over here, and pertussis appears 3 times with some weird stars in it. I'll get back to why in a second. And then finally, our toxoid vaccines, we have diphtheria and tetanus, and pertussis, again, hold that thought. And then finally, for subunits, we have Hep B and, once again, pertussis. The reason we have pertussis 3 times here is that the pertussis vaccine has evolved quite a bit in the past 30 years. Historically, pertussis vaccine was a killed inactivated one. And in that formulation, you had whole cells, you had toxin, you had the whole works, and it was formalin inactivated. It worked very, very well. However, it had a fairly nasty side effect profile. It was extremely painful. It caused fevers, but again, it worked very, very well. And just full disclosure, this is the one I had when I was a kid. And yeah, I remember it being a rotten, awful shot. So it was replaced later on with this guy here, which is called the acellular pertussis. And what this guy was a more or less a suspension of a bunch of mixed antigens from pertussis. So Bordetella cells were grown up, and they were lysed open and purified, and so what you were left was kind of this suspension of proteins that was then formalin inactivated and used as a direct immunization. Lower side effects didn't work quite as well, and so then what happened was the pertussis toxin was added to the mix and was given as a toxoid vaccine. That worked better, but not quite as great as this. So a lot of times what's used now is either this alone or this in combination. All right, so starting at about 6 to 12 months, we add flu shots into the mixed if it's seasonally appropriate. You can give infants either the killed flu vaccine or flu mist. Typically, what people tend to do is save flu mist doses because they're more expensive and a little harder to come by for older kids. And a 6 to 12-month-old baby, you'll give the injected and activated one because they, frankly, put up less of a fuss. And they're not given live virus vaccine for flu anyway. However, they are given live virus vaccines for measles, mumps, and rubella. The problem that you could imagine here is that you've got a 12-month window where these babies are completely susceptible to these infections. And that is becoming a dangerous thing. So older kids and throughout childhood, children are given various boosters and multiple doses of all of the previously mentioned immunizations. Starting at 12 years, you can introduce two more. One is the human papillomavirus vaccine. This is a subunit vaccine that's meant to prevent cervical, anal, penile, and head and neck cancers and oral cancers. And the other is the meningococcal vaccine, and this is meant to prevent against Neisseria meningitidis infection. Okay. In addition to the childhood vaccine series, there are additional vaccines given throughout life, but only to specific populations. So if you are looking after elderly patients, there are three big ones to talk to them about. And the first is shingles, the second is pneumococcus, and the third is flu. You always want to make sure that you recommend flu shots for your elderly patients. If they get influenza, they almost always get this afterwards, pneumococcal pneumonia. And this is how an enormous number of elderly patients eventually die. There's usually underlying circumstances, but pneumonia is an enormously common cause of death for elderly patients. In addition to that, shingles are common in that population, and that goes back to that idea if you can prevent it or lower its duration or lower its severity, that's a critically important thing to do. Another group to be careful of are those with disease complications. The most classic examples to discuss here are asthmatics and hepatitis C patients. If you have an asthmatic patient, pediatric or adult, you had best recommend they get a flu shot every single year. Asthmatics are far more likely to have long-term or potentially fatal complications from influenza infection. So this is absolutely critical that your asthmatics get flu shots. If you have hepatitis C patients, it is critical that they do not, on top of that, get hepatitis B or hepatitis A. And both of those are vaccine preventable. So if you are treating a hep C patient, at this point, many of them have, if they are my age or younger, they will have probably had the hepatitis B series as just a standard of care. However, hepatitis A is less common. Typically, it's only given for people who are traveling to Hep A endemic areas. And so if you have a hep C patient, you best make sure they don't become infected to add insult to injury on top of their hep C infection. The next group we want to, sorry, I've got misaligned here somehow, but the next group we want to talk about are people with occupational hazards. Big one here is animal contact. So this would be your veterinarian, your animal husbandry people, your taxidermists, your animal breeders, et cetera, et cetera. Zoo workers, that's what I was trying to think of. They have to be vaccinated for rabies. They have to be vaccinated for tetanus. Tetanus is carried as normal, Clostridium tetani, I should say, is carried by normal flora of horses. and rabies is carried by any number of vertebrate animals. If they have this likelihood of exposure, it's critical they remain pre-actively immunized. In addition to that, members of the military or people who work in biohazard research with weapons agents typically have to be vaccinated against anthrax and smallpox. For a time, we were immunizing first responders against smallpox in the 2000s, and I think even as recently as 4 or 5 years ago, we've stopped doing that again. However, one never knows when that's going to kick back up. The next group to mention are travelers, and again, I apologize for this poor alignment here, but the immunizations you typically see recommended for travelers, depending on where they're going in the world, would be yellow fever, meningococcus, and typhoid. Yellow fever is typically found in the tropics, and so if you are going to an endemic area, it's critical that immunization is given. And again, it's tropics in both hemispheres. So we have it in Central and South America. We also have it now there's a huge outbreak going on in North Africa at the moment. It goes through Central Africa and Southeast Asia. Meningococcus, it's something that, again, we do routinely now for patients who are 12 years old. However, that was not always the case. And so middle age to older adults have very often not been immunized with the meningococcal vaccine. If you are going to Central or Northern Africa, that's lovingly referred to as the meningitis belt. And so that's an important immunization to include. Typhoid fever, if you're going to any number of endemic countries in the world, which again tends to be tropics, but in some cases further north in both hemispheres. It's when I say further north, it's typically China and India, where you see a lot of this that are not exclusively in the tropics. There are 2 typhoid vaccines. One of them is live attenuated, and this is oral. One of them is killed, and this is an injection. The oral is actually quite easy to take in that it comes as freeze-dried bacteria in a capsule, so it's actually a pill that the patient swallows as opposed to a bizarre drink or anything like that. And as mentioned above, but didn't want to put it on the same slide twice, hepatitis A also belongs in this category. And then the final at-risk population that gets special immunizations are pregnant women. Pregnant women usually get flu shots for the simple reason that pregnancy as a condition tends to be fairly immunosuppressive. People don't usually think of it that way, but it makes quite a bit of sense if you think about it, right? You've got something growing inside pregnant women that is an adorable mix of a parasite and a tumor. At the very least, it's got a lot of non-self antigens. And while the immune system doesn't typically cross the placenta very much, with the exception of individual antibodies, there is a tendency for women's bodies to be quite immunosuppressed. And so flu can be very, very complicated in pregnant women. So it's best if they get a flu immunization. Rubella is something that, again, most American women have been immunized for, and it has, as we discussed earlier in the year, been eradicated from North America. However, that again could change at any moment. But rubella is a virus that causes extraordinarily mild illness in most adults. However, it's detrimental for developing fetuses. So you want to make absolutely certain that any pregnant patients are seropositive for rubella, and if they are not, then you should immunize them. The final one to bring up is pertussis, it's very important that women be immunized for pertussis late in pregnancy. The reason for this is that adults very often harbor Bordetella pertussis, or I shouldn't say very often, but it's not uncommon for adults to harbor Bordetella pertussis in their nasal cavity or their oral cavity, and it's not bothering them at all. It doesn't cause them any symptoms. However, if they transmit that infection to a newborn baby, that baby is very likely to become clinically symptomatic of pertussis, and which is also known as whooping cough. That is extraordinarily detrimental to young infants and small children, for that matter. It's got a respectively high mortality rate, and in those children, infants and children who do recover, they tend to have long-term damage to their respiratory tract. And so things like long-term running and sports and even playing musical instruments, they tend to not be options for those children. And so this is a disease that it's very critical that we get, get under control. The reason why I point out what type of vaccines we're looking at here is that if you spend any amount of time on mom message boards, such as I do, one of the things that you hear with some degree of frequency is women talking about the idea of not wanting to be shot up with poisonous things during pregnancy. And I appreciate the sentiment. However, the reality is it's critical that we illustrate that these are not living organisms, and this is a toxoid, yes, but it's not an active toxin. It's not, it's fixed with formaldehyde, but the shot itself has had 99.999999999% of the formaldehyde removed. There's more formaldehyde on the surface of a pear than there is in that shot. And it's critically important that we really put that in perspective. Not just to say, this is safe, this is effective, don't worry about it, but to say, here is what can happen to your infant if they contract pertussis from you, and you may be colonized and you would not know it. And so it's very, very important that we're as clear and as good at educating our patients as we possibly can be at the risks that they're undertaking. All right, so we mentioned a few times there are some vaccines that are on the horizons, and by on the horizon I mean they're either in clinical trials or have already been approved in other countries. And here they are. So here are the two DNA vaccines that I mentioned, West Nile virus and Zika virus. So the Zika virus vaccine, which as you could imagine has been developed extremely rapidly, this is in phase 1 clinical trials, meaning it's being tested for safety right now. The reason it has not yet, I should say, been tested, it is not yet being tested for efficacy in humans. It has been shown to be effective already in primates, which is promising. The other reason why people are very enthusiastic about it is that it is absolutely bootlegged from the approach taken for West Nile vaccine, which is in phase 3 trials, which seems to be safe, it seems to be effective, and now the trials that are being finished up are trials to using tens of thousands of patients as opposed to a couple hundred or even up to 1,000. The same recombinant, the same protein was used as the target. So, or I should say the homologous protein between Zika and West Nile was used as the target of the vaccine. So the expectation is, because the West Nile virus vaccine seems to work so well, that there's no reason to expect the Zika one wouldn't as well. And in addition to that, the primate studies were completely safe and effective. So it is plausible that we will have our first approved, routinely used DNA vaccines in the next couple years. So, also on this list, we have our recombinant vaccines. We have an Ebola virus vaccine and we have a dengue fever vaccine. Both of these are chimeric viruses made off of this backbone. This Ebola virus vaccine was fast-tracked into clinical trials overseas. The trials were done in Guinea, and this vaccine seemed very safe, very effective, and by all accounts, the patients who received it had almost a dramatic. The patients who received it had a dramatic drop-off of their risk for infection during the 2014 Ebola outbreak. It's thought that ring vaccination with this instrument, coupled with all of the other measures that were taken, was something of a player in cutting short that outbreak in Guinea, at least. The dengue fever vaccine is also chimeric, same backbone. There are 4 serotypes of dengue fever, and this vaccine formulation is sort of a mixture of all 4. And it has been licensed for use in Mexico, and so far it seems to be doing very, very well. It seems to be safe and it seems to be effective, which is great. The last 2 I'll bring up are in their last phases of trials, and they are both toxoid vaccines. One is to prevent C. difficile colitis. It's also called pseudomembranous colitis. You may have heard this referred to as C. diff. This is a really, really rotten infection, and it's something that patients tend to suffer with for quite some time, and it's hard to get rid of once you have it. So it is also a completely toxin-mediated pathology. And so that being said, it was an attractive candidate for a toxoid vaccine. Some smart guys developed some, and they again seem to work nicely. So I look forward to this being routinely used. Similarly, there are development of vaccines for streptococcal pharyngitis or strep throat. Same deal, that we have a bunch of pore-forming toxins that are made by strep pyogenes, and they're called streptolysins. And some smart folks took these guys and chemically inactivated them, created immune responses to them, and severely cut down the damage caused by strep throat. And so hopefully we will see this too someday soon. All right, so the last thing I want to talk to you guys about is vaccine lapses, and I'm going to be as brief as possible here because I think we all know or can guess what happens with vaccine lapses. Disease follows. And there are 2 different worldviews and 2 different ways that this can happen. And the first, to me, is very, very tragic, and that's when you have vaccine lapses that are secondary to collapses in infrastructure. We talked about something that's in a way sort of analogous in our last class together when we were talking about parasitology. And we talked about how, due to collapse in infrastructure in Syria, that there stopped being municipal pest control for sandflies, and predictably when that happened, there was an outbreak of leishmaniasis, and that you now have all these patients that you didn't have before because the infrastructure was successful in preventing that infection. The same thing can happen with vaccination campaigns. When you have lapses in infrastructure and lapses in healthcare systems, you have drops in vaccination rates, and then you have a susceptible population emerge, and there you go, you get disease. The 2 that I've highlighted here are polio in Somalia and Syria, and diphtheria in the Russian Federation. So this is still an ongoing problem, particularly in Somalia. A couple of years ago, no, that's not true, probably just about 1 year ago, there was an outbreak of Syria and polio. Other way around, outbreak of polio in Syria. And the thought was that this was an introduction into Syria from Somalia. And so that this went from over here to over here, and sorry, I overshot Syria a little bit on that, but and that since you had such a collapse in infrastructure in Syria, in addition to the ongoing collapse in Somalia, that what ended up happening was you had a lot of unvaccinated children, because this conflict has been going on in Syria for the lifespan of a lot of children. When you think about it, I mean, it's been going on for years. And so these kids have no immunity to polio, and fighters that emigrated from Somalia ended up seeding the area with polio. And so a number of children wound up contracting it. There's still a bit of activity ongoing in Syria, but it's not as prevalent as it was a year or so ago. That being said, it's still quite prevalent in Somalia, and in addition to that, it's not been unusual to see satellite cases in Ethiopia and even in Kenya. The issue that tends to happen as well with saying that, oh, something is restricted to, this is only a problem in Somalia, is that you have a lot of international border issues. So I love this picture. It's a picture from National Geographic, and it's about something totally unrelated, but the point here is that this is the Somali-Ethiopian border at one spot, and it's a rope. And the reason I love this particular picture so much is that this dude is in the process of actually stepping over the rope. And it just goes to show you that if you wanted to quarantine that border to keep potential polio patients out of Ethiopia and out of Kenya from Somalia, it's challenging because you have this issue of a somewhat porous border. So what Ethiopia had done in years past, and I believe they're still currently doing, is that they had, if this were the border, they had a little shed right about here where they had volunteer aid workers that were immunizing people with the oral polio vaccine as they stepped over the border, in an attempt to just keep it out of Ethiopia to the extent possible. But it's an ongoing issue. And it's a problematic one from the standpoint of there's a narrative that we don't have these diseases anymore. And that simply isn't true. And it's in order to completely get rid of them, there's an incredible amount of societal work and infrastructural work and healthcare system work that would have to go into it. And so it's not simply a medical problem. It's a societal issue. So the other really nifty example of this is diphtheria in the Russian Federation. It's important to point out that this is no longer an issue to the extent that it was, but it just shows very nicely how collapses in infrastructure yielding collapses in vaccination lead to disease. So this down here, over the course of several years, is the prevalence of diphtheria in Russia, in the Soviet Union. And right here, the red line is the collapse of the Soviet Union. And when that happened, there stopped being a central government in place for certain areas of what had been the USSR and were now their own independent countries. And the country of Russia itself was kind of in a tumultuous state. And so things like state-sponsored vaccination programs fell by the wayside, and predictably, cases of diphtheria did this. Once everything kind of regained control and structure, they then did this again. So that being said, it's country after country, society after society, disease after disease. You see very much the same pattern. When infrastructure collapses, vaccine rates goes down and disease rates go up. So that's one way we have vaccine lapse. Way is this. We have, to an almost alarming extent in this country, vaccination lapses due to elective omission of vaccines. As one could imagine, I have strong opinions about this, but I will leave you to form your own. Elective omission is almost always done in a well-meaning way, I think, but it ultimately has a consequence. And as we saw on the previous slide, when vaccination rates lapse, disease rates go up, and that's just how that goes. And so what we have particularly seen problematically in this country is measles and pertussis. And so I've got over here the number of measles cases by year since 2010. This is the CDC census. 2016 obviously is not finished yet, but that's where we are right now. And 2012 was a good year, but other than that, everything's going up, 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 up. This was the infamous, this was the New York outbreak, and then from 2014 to 2015 was the Disneyland outbreak. It's funny, something that should be so very unusual, we're seeing so many of that we can kind of refer to them notoriously by name. It's important when we're discussing measles to be sure that we communicate a few things very loudly and very clearly to our patients. Never mind that the measles vaccine does not cause neurological complications. The word neurological is here, but that refers to something else I'll get to in a second. It does not cause autism. It does not cause seizures. It does not cause whatever else it's being accused of causing right now. But it's best to not even bring those things up when you're talking to patients. There's a lot of great evidence that suggests even when you repeat a misconception in the negative, in other words, saying measles vaccines do not cause autism, you're still repeating measles vaccines cause autism, but with one extra word in it. And so psychologically, that does not register. As a scientist, it didn't make sense to me, but the data are pretty persuasive that that's actually what happens. So who am I to say? I think it's critical we don't even talk to our patients about that unless it's brought up by the patient wanting clarification. But we can say that now hundreds of thousands of children have been enrolled in clinical trials, in retrospective and prospective studies. There is no linkage between autism and MMR shots, just absolutely none. However, what we can say to our patients is that measles is not a simple, harmless childhood infection, that measles has a fairly high risk of complications. We learned earlier in the semester that we can see measles-associated pneumonia. But in addition to that, something we hadn't yet talked about is that you can get measles-related encephalitis. So measles virus, rubeola virus, I should say, can invade the central nervous system. It can also cause permanent hearing loss. And in addition to that, measles rashes themselves can leave a bit of scarring. It's an infection that doesn't need to happen. And It's an infection that can be fatal, and if it's not fatal, can have long-term permanent consequences. That is something we need to say to our patients very loudly and very clearly, because this should not be happening when we have a preventative tool. And the fact that it is happening means that we need to do a better job communicating why we feel it's important that this immunization be taken. I know there are other people doing a lot of communicating with our patients, and those people communicate very emotionally and very persuasively, but we need to be more persuasive, because this is a thing and it should not happen, because the patient in question is a child who has no say in the matter. Even more importantly, I think, is the discussion around pertussis. As we mentioned, pertussis has a rather high mortality rate, and pertussis has serious long-term consequences. So this is an image of an infant with pertussis, and the issue here is that one of the most tragic things to me is to see a patient that thinks they've made a healthy and good decision for their child, or sorry, a patient's parent think they've made a healthy and good decision for their child, and then lose their child to pertussis. Even more tragic is a parent who brought their newborn baby in for a weight check, and their baby got coughed on in the waiting room by an unvaccinated patient, and then that newborn baby ends up dying of pertussis. That's not fair. And it's not acceptable when we have a good, safe, preventative tool. It's not acceptable. And so what we need to do, again, is it's our job to communicate in an open, in a non-condescending way that there is a serious consequence to elective omission of vaccinations. There's a community consequence, there's a societal consequence, and there's a consequence for that parent's own child. And it's just something that we need to communicate loudly and clearly. So that being said, that is all I have for vaccines. Sorry about the non-traditional format of this lecture. If anyone has any questions, please feel free to email me, and I will see you soon.